So now we're going to take a look at enzymes. We're going to do this before we talk about carbohydrates, lipids, and nucleic acids. So here are a list of questions that you should be able to answer when we're done completely with our enzyme discussion. What are the characteristic features that define enzymes? So what makes an enzyme an enzyme? Can the rate of an enzyme catalyzed reaction be defined in a mathematical way? Yes, we will use some equations to calculate the rates of catalyzed versus uncatalyzed reactions. What equations define the kinetics of enzyme-related reactions or catalyzed reactions? We're going to first review with some problems from Gen Chem 2 before we actually start working with our kinetics equations for enzyme catalyzed reactions. What can be learned from the inhibition of enzyme activity? We'll look at three types of enzyme inhibition. How can they be so specific? So enzymes are very specific to their substrates and subsequent reactions. Are all enzymes proteins? We'll find that they are not. And is it possible to design an enzyme to catalyze any desired reaction, or perhaps design a substrate to inhibit a reaction? So enzymes are powerful catalysts. So what is a catalyst? A catalyst is a molecule, or in this case a protein, that increases the rate of the reaction and is not consumed in that reaction. So they are powerful catalysts. We will be calculating the catalytic power, and that is basically the ratio of the catalyzed rate of a reaction to the uncatalyzed rate of the reaction. The large rate enhancements of enzymes range from 10 to the 7th to 10 to the 15th. So these correspond to large changes in the free energy or the activation for the reaction. So we'll be looking at some energy diagrams. All reactions pass through a transition state in that pathway, and we'll learn to represent that using an equation and also identifying that transition state on those energy diagrams. The active sites of enzymes bind the transition state of the reaction more tightly than the original substrate. And what this does is stabilize the transition state and lower the activation energy of that reaction. So just some additional information on the classification of enzymes. All enzymes are classified and named according to the reaction that they catalyze. There are 6 classes of reaction, and those are shown here. The classes of reactions that are recognized: oxoreductases. So, as the name suggests, these catalyze redox reactions. Transferases, transfer functional groups. We'll see this in a couple of our metabolic pathways. Hydrolases, hydrolysis reactions. Lysases, group elimination to form double bonds. Isomerases, basically the rearrangement of atoms within a molecule. And ligases, bond formation coupled with ATP hydrolysis. We'll definitely see this in DNA replication. So these are some examples of the rate enhancements of enzymes. Aromatidine 5' monophosphate decarboxylase. This enzyme catalyzes an essential step in nucleic acid biosynthesis, or the synthesis of nucleic acids. It catalyzes the decarboxylation, I have a hard time getting that out, of OMP, or aromatidine 5' monophosphate, to form uridine 5' monophosphate, or UMP, which is a ribonucleic acid. This column shows the uncatalyzed, or the half-time for the uncatalyzed reactions. This is 78 million years. The uncatalyzed rate, these are first order reactions, so we have a rate of 1 over s, or 1 over seconds, 2.8 times 10 to the negative 16. When this enzyme catalyzes this reaction, that rate is 39 to the 1 over s, which is a 1.4 times 10 to the 17th rate enhancement. So again, this rate enhancement, or the catalytic power, so to speak, is the catalyzed rate divided by the uncatalyzed rate. And we'll do some problems in which we calculate these. We were working with chymotrypsin in our amino acid protein sequencing determination problems. 20 years. Uncatalyzed, 1 times 10 to the negative 9. Catalyzed, 190. That's a 1.7 times 10 to the 11th rate enhancement. We will see triose phosphate isomerase in the glycolytic pathway, 1.9 years. 4.3 times 10 to the negative 6. Rate enhancement, or the catalyzed rate, is 4,300. Huge. So imagine we've got this enzyme in the glycolytic pathway, or glycolysis, that catalyzes a particular step in that metabolic pathway. If that wasn't present, we would not be metabolizing glucose at a rate which we could survive on. So this actually probably should have been moved before that last slide. Sorry about that. Should have followed the classes of enzymes, again, of which there are 6. There's an enzyme commission, believe it or not, and this is the systematic way in which they have come up with naming these enzymes or classifying them. They have, actually not shown here, 4 different numbers that ID what that enzyme is. So here, this is the class, obviously. These are oxoreductases. The 1 here is showing that this acts on CH bonded to hydroxyl groups. We had another one. We have NAD or NADP as an acceptor, and you'll understand the meaning of that later. I will probably have you look up an enzyme and also give me the classification, but we'll talk about that later. So with respect to enzyme specificity, there are two hypotheses. One is now more universally accepted than the other. However, the first theory or hypothesis was put forth by Emil Fischer in 1894. And in this model, both the enzyme and the substrate possess specific complementary geometric shapes that fit exactly into one another. So while this explains enzyme specificity, it doesn't explain the stabilization of the transition state that has been observed and is achieved in that enzyme catalyzed reaction. In 1958, Daniel Koechlin suggested the induced fit model, and in this model, which actually provides a more accurate description of specificity and covers that transition state, the enzyme has a rather flexible structure, and the active site is continually reshaped by interactions with that substrate. So the conformational change in the site stabilizes that enzyme substrate complex. So induced fit is more highly recognized or believed today. So we know enzymes always have a substrate, and they all selectively recognize those substrates over other molecules. Doesn't mean we can't trip them or trick them, and we'll talk about an example of that. We know that catalyzation of that reaction produces very high yields of products. That's greater than 95%, which actually is quite amazing. That type of product yield in a typical lab rarely reaches such a high percent. Again, that specificity is going to be controlled by the structure, the unique fit of the substrate, with the enzyme controls the specificity for the substrate and that product yield. And as you can see by this picture, this suggests we're going to have some hydrogen bonding. We have a NH here, we've got a hydrogen here, hydrogen, which can interact with an oxygen here. So there are going to be interactions between substrate and enzyme that are stabilize their interaction. So, although many enzymes carry out their catalytic function by relying solely on their structure, there are many other enzymes that require cofactors. So we're going to run through this. Cofactors may be metal ions or coenzymes. Many coenzymes are vitamins or contain vitamins in their structure. Coenzymes are usually actively involved in the catalytic reaction. So availability of substrates and their cofactors determines how fast the reaction goes. The enzymatic rate often slows down as the product accumulates and equilibrium is approached. Some enzymes are what we call regulated by feedback inhibition. So they're inhibited by the increasing amount of product that they're generating. Genetic regulations. So the amount of enzymes synthesized by a cell is going to be determined by transcription and then, of course, translation, since it's a protein. So the amount of enzyme present at any given moment also is going to, determine enzyme activity. And that, remember, that gene from your biology can be induced or repressed depending upon what's needed or what's not needed. There may be effector molecules that bind to sites other than the active site that allosterically regulate enzymes. So perhaps an enzyme is regulated by one of these effector molecules binding a different site on the enzyme, which causes a conformational change and inhibits that enzyme's activity. Enzymes can be regulated through covalent modification, and we will see examples of this in some of the metabolic pathways. These modifications are typically phosphorylation or acetylation. Zymogens, which are inactive enzyme precursors. Isozymes, enzymes that differ in an amino acid activity, but catalyze the same reaction. So these are some of the factors that influence enzyme activity. Here's some enzyme or coenzymes and cofactors that are non-protein components that are essential to some enzyme activity, not all, of course. We have metal ions that can be required for activities. So here, Fe2+, Fe3+ for this cytochrome oxidase, Fe3+ for this catalase. We've got copper ions, magnesium. So magnesium ions are required for hexokinase, glucose 6-phosphatase. We'll see this in the glycolytic pathway. We'll also see pyruvate kinase in the glycolytic pathway, and some of these others in other discussions of metabolic pathways. Coenzymes, thiamine pyrophosphate. We'll see this also in one of our pathways. FAD or flavonoadenine dinucleotide, so pyruvate dehydrogenase, succinate dehydrogenase. We'll see these enzymes in some glycolytic pathways as well. So what are kinetics? Basically the rates of enzymatic reactions, and that is what we're going to be interested in looking at right now, the biological roles of enzyme catalysts and their mechanisms. And we can look at these in a number of different ways. Enzyme kinetics seeks to determine the maximum reaction velocity that enzymes can attain and binding affinities for substrates and inhibitors. We will look at these in a mathematical way. Kinetics also analyzes enzyme rate yields, and this can give insight into enzyme mechanisms and pathways. Pharmacology, drug design. We can use information that we gain from kinetics to control and manipulate some of the metabolic events that create problems, perhaps. We're going to look at these terms. Actually, we're going to take a break in just a minute or two, and we'll define these terms together in your workbook. So I'm not going to go over those at the moment. We'll come back to that. I do want to run through a quick review of kinetics before we take that break. So we're going to be looking at first order reactions only. So the simple representation of a first order reaction is A going to P, P being product. So we have our reactant being converted to product. This is a function of time. The course of the first order reaction is a function of time. We describe these as being unimolecular. There is one reactant that goes to product. Bimolecular, we would have two reactants that go to product. And when we start working together in a moment, we'll also give another example for a bimolecular reaction here. Kinetics do not provide reaction mechanisms. We can only determine rates. However, we can rule out alternative hypotheses based on kinetics when the data don't fit. So this is a graph of the time course of a first order reaction. On the y-axis we have the percent of A or R reactant remaining, and on the y-axis we have time. The half-life equation for a first order reaction is T1/2 equals 0.693 over K. And again, when we work together, in a few moments, I'll give you that equation again, and we'll look at some other equations necessary to study or calculate the decay or the rate of a reaction. And actually that's right now. So let's take a break and work some problems together, and then we'll move on with further discussion of enzyme kinetics.